The study guide review. As I told you before, we're not gonna try and relearn the whole thing, okay? It will be more of a what you gotta focus. So you have to know which microorganisms we study, how we call the branches. I provided you a list, and you have to know the difference between them. I mean, which microbes are unicellular, which can be multicellular, which are prokaryotic, which are eukaryotic. What is the difference between viruses and everything else? Why viruses are so much different? Does that make sense? Okay. Remember we discussed why bacteria and archaea are exquisitely prokaryotic. They only unicellular. You have to know why they cannot form multicellular organisms. Signaling, right? Okay? Any questions on that specific question? Not really. Okay. Role and importance of microbes in nature, industry, and healthcare. So, I pretty much gave you answers. Not that I'm going to ask you which of the following industrial processes is mediated by propionibacteria. Okay, no. But have an appreciation to that. Be able to give me examples of how we use them, at least in the food production. Okay? Where can we find them? Well, in the environment it's kind of boring because you can find them everywhere. Okay? In infectious disease, because that's the focus of the course. Understand the diseases that can be caused by microbes and diseases that are not associated with microbial infections. Okay? Classification. 5 kingdoms in Whitaker system. Can anyone name 5 kingdoms? Animals, fungi, plants, Monera, and protists. Okay? Animals, plants, fungi come really easy. Okay? Monera and protists, think about they are all unicellular. Monera are prokaryotic, protists are eukaryotic. What they are based upon. Whitaker system, what is based upon? Evolutionary relationship as we understood them at the time. Woese-Fox system. It divides all living organisms into what? 3 domains, which are archaea, bacteria, and eukarya, right? Understand the phylogenetic relationships between the domains. Which 2 domains are closer to each other? Archaea and eukarya are closer to each other than both of them to bacteria. Does that make sense? Now, what Woese-Fox system is based upon? Genetic relationships, okay? Genetic similarity. 4. That's probably the easiest. First word is genus, second word is species. Well, technically, I mean, when we say staphylococci, we talk about species. We don't say, oh, I've got aureus. No, you've got Staphylococcus aureus. Okay? It's still 2 words for the microbes. The first means genus. When it's combined with the second, it gives you a species. Major milestones. May I skip on that? You have a table. Huh? Now look. I I may ask you things like, who was the guy who proposed postulates linking infection and the disease? You must know that it's Koch. Okay? In that list, you have to refer the name and the contribution. You can make a set of about 15 flashcards. It's going to take you an hour. Really, it's maybe less than that. When you will finish making the flashcards, you will know all of them. I'm dead serious. Okay, so it's fairly straightforward, and we're going to come back to them. Structure of the water molecule. Well, know that it's polar, that it forms hydrogen bonds between positively charged hydrogen and negatively charged oxygen. Understand why hydrogen bonds are important. They determine the shape of macromolecules like DNA or protein. Question? No? Good. pH scale. I'm not asking you to tell me that tomato juice has pH of 4 and Coca-Cola has a pH of 2.5. I don't care. 7 is neutral, above is basic, below is acidic. That's it. Good? That's it. That's really it. Now, carbs, I'm going to talk about all biological macromolecules together. Okay? Carbs, fats, proteins. Okay, carbs. For all of them, for carbs, for fats, for proteins, understand that dehydration, taking water away from basic units like monosaccharides, will give you a polymer, a polysaccharide, right? So then, in order to break polysaccharide in pieces, you have to add water. You have to hydrolyze them. Okay, dehydration is synthesis, hydrolysis is a breakdown. Clear? Know the function of all of them. So for carbs, it's going to be energy and structure. Lipids. Know the components of triglycerides, glycerol, and fatty acids. Phospholipids, phosphate, glycerol, and fatty acids. Sterols and hopanoids. For sterols and hopanoids, it's hydrophobic, large hydrophobic compounds. I'm not going to ask you to draw the picture. Okay. Function. Triglycerides, energy. Phospholipids, membrane. Sterols and hopanoids, membrane rigidity in eukaryotes and prokaryotes, respectively. Does that make sense? Again, dehydration, hydrolysis, production of triglycerides. Okay. So when you take free fatty acids, let me do this. When you take free fatty acids and glycerol and take water away, you get triglycerides, right? When you take triglycerides and add water, you break it in pieces into free fatty acids and glycerol. Cool? Terrific. Proteins. Know the components. I get really mad when you pick that proteins consist of nucleotides, so don't. They consist of amino acids, okay? Dehydration, reaction of dehydration, amino acids form peptide bonds and form proteins, okay? Proteins have 4 levels of structure. Understand those levels. Primary is the sequence of amino acids. Secondary is alpha and beta sheets. Tertiary is alpha helices and beta sheets that are folded together. Sometimes several protein molecules form quaternary structure. Understand the idea of denaturation. when we change the environment, when we add acid, base, heat it up, do something else, proteins can lose the structural level. That's denaturation. Does that make sense? I'm making myself clear. DNA. Well, nucleic acid. Structure of the nucleic acid. Base. Sorry about that. Sugar. Phosphate. As components of the nucleotide. Understand the idea of complementarity. Know that nucleotides form DNA and RNA. Know the function of DNA, storage of information. RNA, conveying the genetic information from, to serve as the template for protein synthesis. Okay, and again, dehydration, you've got your synthesis, adding water, hydrolysis, you break down DNA. Anything that is unclear about that. There are like 5 slides, and I'm gonna ask only what is on those slides. I'm asking anything that's on that slide, okay, but only what's on there. Does that make sense? I went through the questions yesterday. I'm pretty confident that I'm not asking anything that we've never talked about. All right? Yes. No. Well, yeah, pretty much dehydration is when you take water away. Hydrolysis is when you add water. But when you add water to carbohydrate, like a starch, you produce monosaccharides. You break down starch into monosaccharides. When you add water to a protein, with the help of some enzymes, you produce individual amino acids. So hydrolysis always causes the breakdown of the large molecule to the building blocks. We cool? Genotype and phenotype. One thing, the most important one. What defines the phenotype? Well, trait is a phenotype. So what defines it? Expression. So can you say that the exact sequence of nucleotides define phenotype? Not really. It's important, but depending on which parts of the genome are expressed at the time, the phenotype will depend on that. Does that make sense? So expression of the genes. Concept and mechanism of evolution in the light of adaptation to the changing environment. It's horrible. It sounds awful. What does that mean? What do you think? When environment changes, what also changes? Gene expression. Phenotype changes. Does that make sense? Mutations also alter the gene expression. They alter the sequence, they alter the function, they alter the structure, they alter the function. So mutations are the mechanism of evolution, of course. Okay? Help to adapt to the environment, but changing conditions change the expression. Underlying principles of microscopy. It's kind of a self-explanatory. Refraction of light. Understand the difference between magnification, resolution, and contrast. Know how we calculate magnification. How we calculate magnification? We have to multiply. Objective times eyepiece, yes. Got it? What limits light microscopy? What is the major limitation of light microscopy? Which physical parameter? Which physical parameter of light? The wavelength, right? The wavelength, yes. or frequency. It's a major limitation of light microscopy. Does that make sense to you? So which parameter wavelength affects? Magnification, resolution, or contrast? Just one. Not really. It's amount of light. Huh? Resolution, of course. Wavelength or frequency is what resolution depends upon. With oil immersion, you use oil immersion because with oil immersion, when you go to the... Now think about this, that's a good question. We didn't really talk about oil immersion, and we didn't try oil immersion yet. So I actually cut out the question on oil immersion out of the exam, so don't worry about this. But it's good that you brought it up. So oil immersion lens is when you use magnification of 1,000 times. In this case, you magnify image to such an extent that when you get image larger and larger, the part of the specimen that you look at becomes smaller and smaller. Does that make sense? So the amount of light that you get becomes smaller too. When you have such a little light, you want to focus it more. Oil immersion, the oil bridge, quote unquote, between the objective lens and the specimen, the bridge that fills up that air gap, allows you to bring in more light. Does that make sense? So it increases contrast rather than resolution. Okay? Major components of the microscope: iris, condenser, light source, objective and lens and eyepiece, mechanical stage, fine coarse focus knobs. They're all obvious. Okay? Probably condenser and iris are a little different. Not so common. Iris allows you to regulate the amount of light. Condenser focuses the light on the specimen. Other than that, everything else is obvious. Really self-explanatory again. Major types of light and electron microscopy and their application. Now what it means is, remember the table, the summary of the types? This type of light microscopy uses I want to say fluorescent, but then it's going to be fluorescent microscopy. You know, I try to avoid the direct clues in the question. Okay, this type of light microscopy takes images slice by slice, changing a focal length, and allows you to produce 3D image of the specimen. You say confocal. Okay? Or I may ask you, what is the unique feature of the dark field microscopy? Use fluorescent antibodies? No. You observe against the dark background? Yes. Does that make sense? So be able to relate the name and the definition. Okay, it's multiple choice for God's sakes. For electron, you gotta know two: transmission and electron, transmission and scanning. Scanning is the surface, transmission is inside the cell. So which microbes generally cannot be seen with light microscopy? Viruses. Everything else generally can. There are some exceptions in both fields, but when I say generally, think about the general answer. Why do we fix the specimen? Why do we fix the specimen, the microscopic specimen? Yes, but why? It doesn't move. Yeah, so it doesn't move. Why do we stain? Huh? How? To increase what? Not resolution, huh? Contrast, right? Not resolution. We can't really do anything about it. But increase contrast. Major types of staining. Simple versus differential. Know the difference, okay? In simple, positive, negative. Staining of the specimen versus staining of the cell versus staining of the background. Differential, the ones that we talked about. Gram, acid fast, endospore, capsule, flagellar. 5 types. Did I make myself pretty clear on that? Okay. Again, I will not ask you the technique of gram staining. You don't have to describe the steps. Okay, but if I ask you, this staining allows you to distinguish 2 basic types of microbes based on the ability to retain crystal violet stain. Flagellar, capsular, gram, endospore. Well, this is obvious. It's a gram stain. Okay. Does that make sense? Any questions on microscopy and staining? Always get a question, how in depth do I have to know? Again, I told you, deep enough to, when you see the definition, you know what it's talking about. That's enough. Okay? That's good. Steps of the culturing. Inoculation, incubation, isolation, inspection, identification. 5 steps. We good? For each step, for inoculation, well, understand the idea of sterile sample collection. Which samples we can take, okay? Types of media. Non-living media versus living media, like cells, animals and eggs and stuff. Types of chemical media. Defined versus complex, right? Differential versus selective. Enriched media. Okay, liquid, solid, semi-solid. Know these classifications, we talked about them. So classification based on the physical state, based on, so I highlighted here, biological form, living versus non-living, physical state, they shouldn't be comma. So biological state, physical state, chemical composition, defined, complex, functional type. Okay, general purpose, enriched, selective, differential. Understand the different types of cultures. What is pure culture? 1 type of microbes. What is mixed culture? What you've got today, many types of microbes. What is contaminated culture? You want 1, you got many. That's contamination. Okay? Why we grow microbes in the incubator? Let's say it again. Why? Why it's important. You're not going to get away that easy. It is controlled environment. Why it's important? Huh? Less variables. Huh? What does it mean, less variables? Okay, so if guy in Cincinnati wants to do the same, he's going to get the same results if he uses our conditions. What else? So replication, well, replicate experiment and also compare. Does that make sense? So if guy in Cincinnati says, I have different results, we ask him, how did you do that? My incubator was at 25. Well, ours was at 37. We can't compare. Make sense? Streak, pour, spread plate, hockey stick, streak plate. Remember, we talked about it, right? Streak plate, when you thin out the culture, right? Loop dilution or pour plate, kind of covered it. Characteristics of a microbe that can be derived from inspection of a culture. What can we say? When we inspect, did you inspect your cultures today? Did you? You did. What information did you get? Huh? Color of the colonies. Size of the colonies. Number of the colonies. Shape? Shape. Even appearance. Like mucoid versus non-mucoid colonies. Identification. I don't know. If I would be you, I would be afraid of that part the most. And it would be wrong. Because my purpose is not to ask you the fine details of how we do, like, describe me the whole PCR method, okay? Instead, when we do PCR, what do we detect? Do we detect the whole DNA sequence? No. We take a little fragment that is unique and detect it. Does that make sense? So you have to understand that. When we do immunofluorescence, what do we detect? We detect an antigen on the cells by using fluorescent antibodies. When we do Western blot, we detect a protein in the mixture that was separated on the gel. Does that make sense? So that kind of a brief description of a method, right? Any questions on identification steps, the whole culturing thing? Spontaneous generation. Know about Redi and Pasteur, those two experiments. Koch's postulates. I'm not going to be asking about postulates themselves, but know what Koch did. Definitely know that those are postulates that linking microbes, the microbial infection and the disease, microbes and disease. Endosymbiosis. Which two organelles we talked about in terms of the endosymbiosis? Mitochondria and chloroplasts, okay? Know why we can see, why we have a convincing proof that they are endosymbionts. Because they have DNA and they can replicate independently from the cell. Okay? Is that clear? Question? Go ahead. Okay. Structures. Coccus, Bacillus, Vibrio, Coccobacillus, Spirochete, Spirillum. Sounds like a handful. Coccus, sphere. Bacillus, rod. Vibrio, curved rod. Coccobacillus, something in between the spherical and the rod. Spirochete and Spirillum, self-explanatory. Spiral. Right? Now, this word, supracellular structure. Supra means above. So, when you have Diplococcus, two cocci, tetrad, 4 cocci. Strepto, chain, like Streptococcus or Streptobacillus. Staphylo, bunch. Okay? Concept of osmosis, idea in the maintenance of cell shape. Well, osmosis, water goes through the semi-permeable membrane from the low towards high solute concentration. That's one. Second, understand what will happen to the cell in the hypotonic or hypertonic environment. Right? Now, what determines whether the environment is hyper or hypotonic? Tell me. Huh? Where? In the solution that surrounds the cell? Okay, so amount of solutes in where? Inside the cell? Outside? Outside only. And inside. That, remember that. Because if I have a brine inside of my cells, then brine is isotonic for me. Remember that. Okay, it's a relative term. Okay? So be able to explain how microbes can survive in hypersaline environments, like environments with a high salt concentration. Remember, they maintain high salt concentration inside of them. Did I make that clear? Okay. Major cell structures and their functions. Everything that we talked about, starting from what are the 4 structures that are present in all bacterial cells? Cytoplasm, ribosomes, membrane, and DNA, nucleoid. Okay, 4. Everything else may or may not be there. But still, you have to know the functions of inclusions, the functions of fimbriae, pili, flagellum, capsule, slime layer. Okay, what we talked about. For cell wall, differentiate the structures of gram-positive and gram-negative cell walls. Good? Good enough? Understand the difference in mycobacterial archaeal. All you have to know that they are different from bacterial, maybe carbohydrates, maybe glycoproteins. That's it. That's what I'm telling you. I'm not asking above the text. Okay, that I provide you. That's why I know sometimes my wife looked at my slides and she said, they so overloaded with the text. And I tell her, look, it's a book. You don't even need the book. Pretty much the information that I give you on the slides is, unless it's something I forgot to put in there, I'm going to write it down on the slide while we're recording that. Okay? Principles of bacterial movement, what we talked about, run and tumble, chemotaxis. Chemotaxis, movement towards the concentration gradient. Does that make sense? I may ask you, I will ask you actually, the transport across the membrane. So know stuff about diffusion versus active transport. Okay, so know the difference. Simple versus facilitated. Diffusion versus active transport. Which one requires energy, which one doesn't? Which one goes along the gradient, which one goes against the gradient? Is that clear? Did I make myself clear about the expectations on the exam? Okay, exam's going to be, I started off with 93 questions. trimmed it down to 50. I will go over that again. Maybe I will trim it down a little more. That's multiple choice. Now, let me pause it.